Can we run it right now for a second?
It's on 4. There we go. Let me switch over to
Okay.
which.
Quickly.
Keep moving, keep
Yep.
moving, keep moving. All right, got it there. And just let me know when you're ready.
Ready?
All right, suction pump on.
So I'm gonna lower it. I'm gonna lower the suction a
All
bit.
right. Around like 1750.
Okay. All right.
There we go, yeah.
Okay. And there's floaty bits in the forward box.
Yeah,
That's probably all we're gonna be able to do, but we're gonna have to just bump it out a little bit.
yeah, just bump it out, and then hopefully the sea cucumbers have settled now, and they won't take off right away.
Yep. Just gonna have to rack back a little bit on the camera. All right, when you're ready, you can bump it out a little bit.
All right, bumping out. That much.
And then you can kill suction.
All right, stopping. Yes. Perfect. That was amazing. All right, closing. Nicely done.
Bravo. That was very impressive.
That was intense.
Yeah.
Nice.
Is this sample 37?
This is sample 39.
39?
Yep.
See if I can get some of this mud off the vehicle.
So we have a comment saying the lucky nudibranch and the team says save the day.
Yeah, indeed. Thanks so much, pilot. Great job. So just as we're getting settled here with the new watch and a time check. So we're at 2,527 meters, so we'll try to make our way up here. At 2,500 meters, we're going to collect a Niskin bottle, and then we're going to keep moving up the slope. Considering that it's going to be a lengthier ascent to the surface, we have a little over 2 hours of bottom time left. We do have to be back up on deck at 4 o'clock. That's a kind of a strict cutoff because we have a busy schedule in the morning.
Meters, but I'll keep coming up as you.
So yeah, order of priority is going to be mostly to cover ground here and try to document with that snap zooms anything that has not yet been documented on this dive. And then the second order priority is securing samples of anything that is of scientific value.
All right, want me to get you moving?
Yeah, let's go for it.
Okay. It looks like we are going 290. Bridge nav. Good morning. We would like to move 40 meters 290. Thank you.
Keep an eye on our tether.
Oh yeah, that's definitely loopy.
Yeah, well,
What do we got for?
we were kind of behind. Do we have turns?
No,
No. All right.
maybe half a turn.
Oh.
Yeah, so for those viewers that might have joined us recently, welcome, Ali. So glad you're with us. We're currently at a depth of 2,500 meters, diving on the eastern slope
That looks a little better.
of Babeldaub Island. This is going to be the closest inshore dive that we'll conduct as part of the Lebus voyage, a 3-week expedition back-to-back expedition exploring deep sea habitats in and around the Palau National Marine Sanctuary. And this is one of the deeper dives that we'll have during this expedition. And we started the dive several hours ago at a bottom depth about 3,000 meters and then started working our way up the slope. We have about 2 hours of bottom time remaining and we'll try to cover as much ground as possible. So thank you so much for joining us.
Those circles are weird. You see that? In the sediment?
Oh, yeah. Trying to clear this sediment while we wait for the
Yeah.
Atalanta to start swimming.
Too bad we couldn't use the suction to, like, you know, just...
Clean it up like a vacuum cleaner. Yeah. I've thought about it.
Seems to be something black on the seafloor over there.
All right.
A viewer is asking, does the silt on the Hercules compromise the water sample?
Well, we're using it for eDNA. So the sediment might have like DNA from things that are living in the sediment, so it might actually be helpful, possibly. But also, it could be challenging to filter. Generally, we don't want sediment, but that's why Jake's trying to clean it off.
Just gets in the way of the pretty picture.
Oh, there's a spiky sea cucumber over there. I think that black thing's just like a piece of wood or something.
Yeah.
Yeah, I'm just pulling up here in the back row some photos from last September when we were diving out in the northwestern Hawaiian Islands where on sedimented seafloor we saw very similar kind of circular mounds and different trails. In the scientific literature, you often have these things referred to as Lebensspuren, the German word for
Go for a zoom there.
trails of life.
The sea cucumber is so cool looking. Look at it. It's a sea pig.
That's a sea pig?
Uh-huh. So this is a sea cucumber in the family Alpidiidae. And see, it's got those little walking legs. This is different from the 2 cucumbers that we have in the box currently.
It is a strange looking one.
Isn't it?
Yeah.
I've never seen this one before.
Can we confirm that it is different than anything we have?
Yeah, so
Yeah.
when we opened the box and the cucumber's trying to escape, we had an Amperima pennyagony and a regular non-spiky synallactid. So both of those are in different families from this one. This is different than the two that we have. Did we slurp any cucumbers, May?
Let's see. We have slurped. I believe we did. Hang on. We have a brittle star, we have an urchin, and crinoid.
There's a fish, and I saw a shrimp.
Yeah, we don't have any sea cucumbers in the slurp.
Okay.
Yeah, so I think if we were set up to do so, we'll probably try to collect a sample here.
Okay.
Okay.
Yeah, and we have a bit of sediment
Thanks,
in slurp number 4, but no sample.
This is not going to fit in the slurp, so we'll have to pick it up with the claw and drop it on the rest of the cucumber family.
Yep. So we can use the flush or 4 in order to.
You don't think it's going to fit in the slurp?
No. See the lasers? It's
Get clogged.
too chunky.
Too chunky?
Yeah.
But you're thinking pick it up in the claws?
I think you could.
Do what you did before with the urchin.
You could either try to suction it and drop it in, or try to just, if you can lock it kind of open.
That's my preferred method.
Yes, suction it.
Rather than crushing it.
Okay.
That's always an option when you're trying to grab it in these snaggletooth claws.
Yeah, the craft claws are crafty.
The coral cutters are hard.
Well, and also with the way that the jaws function, it's a little more challenging.
Tilt down.
Maybe just turn the suction down a little bit more.
We'll
just try it.
Yeah.
We can do a lighter suction, yeah.
Okay. Maybe go like 1,000 PSI and see.
If we suction it too hard, it could.
Yeah. Maybe we should go even less. Could always
This
turn
one looks kind of robust, so it should be all right.
We'll start at like 750.
Do you want some zoom? Are you good?
All right. Let me know when you're ready.
Yeah, 1 second. All right, ready?
All right, suction coming up.
Are we on the right jar? Do you know we're on the right jar?
I think we're
Just
still
in
on the
case?
flush jar.
Just want to make sure we're on the flush. Yeah.
Here, I'll pull that up for you.
Okay,
There you
cool.
go. Yep.
All right. Increasing suction.
Up to 1,000.
A little bit more.
We're at about 1,500 now. Keep going.
All right. That's probably good. It's starting to go.
Okay. Right around 2,200.
There we go.
Nice. Perfect.
Oh, now you can really see its little feet.
So neat. All right.
Doesn't it look like really cuddly?
Yeah, I guess. That wouldn't have been
You
my
know, if you need
first
me to
like,
retract.
Yeah,
adjective
retract.
for it.
Alright.
It just like wants to give you a hug.
Get a little bit closer here.
It's like a cuddly balloon animal.
Like a dog toy.
I bet the dogs would definitely be into that. It would be a fun one.
Can bump out.
All right, box coming out.
Whoa.
That was nice.
Perfect.
All right, can I kill the suction?
Stopping.
Wow.
Yeah,
Well
close
done.
up.
Nicely done. That was great.
job.
That's pretty smooth.
Very smooth, quick and easy.
Nice.
Good job.
Yeah, that's a great collection. Thank you so much.
Thanks.
Yeah, that was a really neat one.
Yeah, that one was really cool. Yeah, I don't know. It's probably not like a Scotoplanes, but it's in the same family. So like Scotoplanes is the like official sea pig, but I feel like all of them that look like that should be called sea pigs because it's really cute.
Tilt up just a little bit more here.
Bridge nav.
Here's one of those.
Can we make a 40-meter move to 90?
Here's one of those rings you were.
Thank you. 40 meters, 290. Yeah.
Yeah, quite a diversity and abundance here of all these burrowing trails. There's quite a bit of information in the literature. Different researchers using these also to quantify of what types of marine animals are there. So some burrowing holes are produced in a very individual manner by different species. And so yeah, these types of life trails or life traces have been used to quantify marine life in the deep sea, and in particular in abyssal habitats. So we're a little bit shallower than that, but
Well, that sea cucumber has a little sail, I think. What's going on?
Sail.
I saw something moving. Maybe it's just another synallactid. Yeah. It was a little piece of debris that was floating. I was like, what's going on there?
Can you come down on the winch 5 meters, Sarah?
Yep.
Yeah, all these animals down here leave little trails. The sea cucumbers have their little cucumber trails, and the urchins have trails, and there's a bunch of animals that bury in the sediment that'll leave different marks on the seafloor. Oh, and there's a sea pen right off to the right there.
What a little.
Yeah.
Looks like a protoptilum.
Yeah, I'd say so.
Go for a quick zoom there.
Excellent,
little
thanks.
sea pen. All right, come wood.
Oh, there's a nice spoke trail. So that one looks kind of old, but there's usually an echiern that lives down in there, and it'll like extend its proboscis and then drag it, so it ends up making this spoke pattern.
What's going on here?
This could be a scar from a shark or a whale coming down to the seafloor.
Scour.
Yeah. I see a shadow
Shadow.
right at the top of the screen. There might be something if you look up. Or is it? Oh, maybe it's a piece of sediment on the camera. Never mind.
Those 2 marks have been there.
Yeah, so for those viewers who have been joining us on this expedition, this is our 3rd successful dive that reached the seafloor, and we're exploring a very different habitats than what we explored previously. So these are more of a soft sedimented habitat, so very different fauna and habitats. A lot of sea cucumbers, which are commonly found on these, that are feet on the sediment and extract things from that. And there, we had 3 successful collections on this dive.
Could we get a Niskin?
Niskin.
Yes.
Want a Niskin?
Yep.
I'm glad, my Connie, that you were watching that as a like a hawk.
Yeah.
Thank
So
you.
it should be Niskin 2.
Niskin 2. Okay.
Right, pan.
Can, yep, rack back the camera and pan left.
Let's go back a little. All right, and pan over.
Niskin 2, you said?
Yeah.
Yep.
Oh.
It went.
There it goes.
Nice.
Yeah, I'm particularly excited about the results of the environmental DNA work. So for those of you who are Nautilus enthusiasts and have been following along over the years, so we have supported several eDNA sampling projects in other regions that had different central focal points. So over the last several years, we've been collecting samples all over the Central Pacific that we're mostly targeting environmental DNA for octocorals, soft coral
I'll
biodiversity.
extend out a little bit.
That was just basically you making use of Niskin bottles on the ROV, and in areas of high coral diversity. So it's work led by Meredith Everett and Steve Alskich, who is on the line with us right now. And then we also have this past year supported work in American Samoa.
Another one of
Working
those
on
cucumbers.
environmental DNA in the water column. And so, yeah, it's really excited now that this more comprehensive work that has already been ongoing
Another
in the
cucumber.
Palau National Marine
Oh, yeah, that's
Sanctuary.
right, one of those.
And yeah, we've been collecting surface samples and now bottom samples, and not just this, but this will add to the ongoing sampling that's happening within the sanctuary. So, yeah, really excited to see how all these samples compare.
See right there.
Oh, yep. Uh-huh. Breaking Megan's rule. But it's for
Well, it's not for, you know.
engineering purposes, let's
Yeah.
call it that.
Yeah.
Technical purposes.
It's a note. It's a reminder that when we get on deck, check that out. Lots of cucumbers. We could make a salad. See, there's a little spiky thing. What is it?
Go for a zoom. I might dust it out in a little bit, but
I think it's an anemone. That's really weird looking.
Is there any way that it's a...
Oh,
1, 2, 3, 4, 5, 6,
It
7,
might
8,
be a
9,
seastar?
Yeah.
10. I got 9. 9 legs. 9 arms.
Well, and one of them looks different than the others.
Oh yeah. Not sure what it is.
I don't know what it is.
It is interesting.
Yeah.
What do you think, Daniel?
Yeah, I think this could be another collection event if we are set up to do so.
Okay.
Alright.
Yeah. Slurp?
Bridge now. Yeah, we should slurp it.
Yeah, let's slurp it. Check out.
Can we hold position?
What jars do we have free?
I think 4 is free,
Okay. I want to get...
right?
For slurps?
Yeah, for slurps.
Yeah, so 4 has a bit of sediment in it, but it doesn't have a sample. 5, 6, and 7 are totally empty.
Okay.
But
Alright, we can go for... Get jar 4 ready. I'll get the arm ready.
We'll rotate, what, clockwise? Yeah. So number 5, we're okay? Oh, wrong way.
Yeah, I think the... Somehow got reversed at some point.
Yeah.
did it have 9
Oh,
arms?
7?
4? If we,
7
Jake-
legs.
Oh, 4.
Jake said 9.
what?
9 arms.
Oh, yeah.
Like-
One of them was like significantly different than the other.
Yeah.
6, we're
Was
at
that weird? It
5.
was almost like
Yeah,
a
it's
tail
like-
with something fanning off the top.
4
Yeah,
has...
it's really
Set,
weird.
set a minute, yeah. We're okay with that, or?
Do you want 4?
To go in 4?
We're
Is
probably going to end up sucking a bunch of sediment up anyway.
So 4?
Yep.
Probably be fine.
Okay.
Excellent.
Yeah. Okay.
And...
Can tilt down on the camera. just a
Yeah.
bit.
Tilting.
Nice.
There we go.
Did you say you've never seen anything like this?
No, I don't know what that is. I'll know better once we get in the lab, but
It stumps me and it's something worth picking up.
my original thought was it was an anemone, but it doesn't look like an anemone.
Yeah, I agree. I don't think so, but
Wait for this little bit of dust to settle. We have to tilt back up on the camera.
All
But
right.
there are a lot of things out there that we haven't seen yet, you know? I might know a lot of things, but I haven't seen it all yet.
Just going to
It's
retract
exciting,
or-
though.
Oh,
Yeah.
tilt
It's
up.
really
Tilt up,
exciting.
yeah.
There we go.
Oh, I see it. It's right over the ledges. There we go.
It blends in well.
Nice.
It does. But I did spot it.
You
so-
did. No, it like
Oh.
blends in.
Oh,
super
it's doing.
Suction?
well. Whoa.
Hold on 1 second.
All right.
Okay, we can go for suction.
Go for suction.
Oh. Whatever it is, it's like, okay, did it go in there?
No, I think it's still there. Do you want me to zoom in?
Yeah, can you zoom in?
Because I think that's it.
Oh,
right
yeah.
there,
Oh, yeah. It just blended so well. Oh,
Oh,
There you go.
it looked like it's had something.
In the sediment.
Yeah.
Okay.
that was weird. All right.
Let's watch the jar.
It might be really hard to see. Oh, it's in
Oh,
there.
it's in there. It's in there.
Yeah.
All right, stopping.
What is it? It looks like it's got like a little peduncle kind of situation. All right, well, I'm excited to see what it is.
That's cool.
Likewise.
Yeah. Can you come wide, Aaron?
Yep, I can.
Thanks. And then tilt back down on the camera a bit.
All right. Yeah.
Yeah, and I'm also, I think this is a really good example. So I think a lot of the dives of deep sea exploration here over the last several years in the Pacific, of various programs have focused on hard bottom substrates, and a lot of them have just focused on habitats that are typically dominated by corals and sponges. So on this particular expedition in the planning process, we decided to focus on a wide diversity of habitats, including soft sediment communities, because you find very different animals and things. And yeah, already on this very watch, just an hour into it, I've already seen quite a few animals I've never seen before. So yeah, it's really good sign.
Yeah, that's a good point, Daniel. Like on our first dive, I was like, yeah, I know what all these animals are. And I'm like, oh, I haven't seen that cucumber. Like, what's this thing? It's been great. Sometimes we think the sediment is a little boring, but the more you look, the more you see. And there is an abundance of life inside the sediment. Even if you're just seeing trails on the sediment, that's indicative of a lot of activity, even if we can't see these really big charismatic megafauna.
Okay, coming back under the vehicle.
Yeah, exactly. And then there's this trend just, globally and historically, and when you look at just the scientific approaches, so there's usually a big focus on, the charismatic and big animals and habitats, and some of these things that are a lot more representative of what the large scales across the deep sea look like, don't get a fair share of the attention, and they are just as important to study, and a lot of biodiversity here that's just not as visually, but as you can see, there's traces of life all over. So within this sediment, there are thousands, if not millions, of animals. And if you want to understand about how patterns on life, patterns on Earth exist, you have to study all of the environments. Yeah, you can see all here the deep sea highways, trails of all kinds of animals.
It kind of looks like the surface of the moon. sometimes. There seems to be a lot of bioturbation here where you see dark patches and light patches. It's indicative that there's like a lot of movement inside the sediment. Bridge, Nav. Can we move 40 meters 290? Thank you.
Some of our scientists ashore, Asako and Chris, are chiming in, thinking that it could be a sea cucumber or perhaps a Scleractinian.
Yeah. I mean, I had those guesses too in my mind, but I just was like, well, those are very different things.
Yeah. Yeah. I was thinking sea cucumber.
Yeah.
Over seastar, but
It was definitely feeling very echinoderm-like, but when it had that, like, peduncle down in the sediment, that kind of felt different than what an echinoderm would normally do. So it could be a Scleractinian, because there are Scleractinians that do grow in sedimented areas. We will find out when you get in the lab. I think it should be more obvious.
Little thing, it was like at the end of the trail, to the bottom center of the screen.
Sea cucumber.
That's a sea cucumber? Cool.
Mm-hmm.
It's like has a trail behind it.
Well, you know, they're going places, doing things. They're busy.
Living their best life.
Is this the place to be if you want to be a sea cucumber? Sarah, didn't you say you wanted to be a sea cucumber earlier?
Say that again? Sorry.
Didn't you say you wanted to be a sea cucumber earlier?
Yeah.
This is where you would hang out. Look at how many friends you'd have.
And all of a sudden it would be like aliens came and sucked me up. As every time I see like something get slurped, in my mind, I'm just like, it's just like a water slide. Like, it's fun.
There's another one of those urchins we picked up.
Uh-huh.
It's like, welcome to watch, Jake. Pick up this like really crazy animal.
so our scientist ashore, Chris Kelly, looked back at the video, and due to that little ornamentation on the end of one of the arms, is thinking it's a sea cucumber indeed.
Okay. Well, I'll trust Chris.
Yeah.
Yeah,
I
that
would
weird
too.
little different modification was
Yeah.
strange.
So what was it?
Cucumber,
Cucumber.
Is the leading guess.
I would have no guess as to what family that belongs to then. No idea.
This cucumber with a bunch of legs.
I've never
Arms.
seen one that looked like that.
Legs or arms? Both?
They're kind of not really either of those things. They're like weird projections. Pseudopodia.
Pseudopodia.
But they do like pump water through their body so they can move their little projections in very specific ways.
Huh.
There's a something floating.
Floating?
Yeah, just coming middle, scream upper.
Oh.
Oh, little fish.
Might be at the end,
Or it
end
could
of
be
the
a shrimp.
tether.
It's giving shrimp.
It's so skinny.
Go for a push in there. Might be a little bouncy after I get to the end of the
Oh,
tether.
look at that. Great job.
Shrimp.
Shrimp. Shrimp.
Nice.
So graceful.
They are.
I was watching the monitor and it looked like the other ship came across one that was like super red, but like the bulk of the front of it was like a deep purple.
Mm.
It was beautiful.
Yeah. Oh, is that the one that was sitting in the
Right.
sediment?
As far as I can chase it.
I feel like this one was moving,
I'm coming.
what I saw. All these circles.
Bridge, Nav. Can we move 40 meters to 90? Thanks.
290.
Yeah, just a little bit of context here, particularly for our followers that are not from Palau. So I just plotted where we currently are. So we're actually just about 3 miles offshore, so right from the edge of the reef where the wave breakers are, and we're currently at 2,500 meters. And if you go just 12 miles east of us is actually where the dive reached the deepest part of the Palauan Trench at 8,040 meters. So it's an incredibly deep, steep slope here. So going over 8,000 meters deep and just about 15 miles. And so yeah, we picked this area of Palauan specifically because it is the place where you have the steepest gradient there. Were several dives in the trench, and then several drop camera surveys just inshore of where we are. And so this dive was basically trying to bridge those two previous
Let's
surveys.
look at the stalk.
Stalk.
Yeah, it's like bottom screen.
Okay.
Just one out of view. There's something on it.
push in there.
It looks like a gooseneck barnacle. And a cereanthid, perhaps? Bunch of hydrozoans. Very cool. It's like, here's the one hard substrate, and everybody's living on it.
Yeah, it's a really good illustration. So if you were to put tiny current meters on the sediment itself, and then at elevated places, different heights above the seafloor, you would measure basically absolutely no currents right at that interface of the sediment and the water. And then as you get farther away, you would have an increase. So there's this benthic boundary layer where there's absolutely no flow, and for things to feed, even those things that are in the sediment, they have to get off up the bottom, and you have a lot of worms that will basically have appendages that will put their arms up into the water and where there is flow and things can be captured. And as Megan alluded to, yeah, everything will try to get us far up above that benthic boundary layer where there's no flow and it's very difficult to feed.
Oh, the barnacle's coming out.
Oh,
You can see its little legs.
That's great, thank you.
all right.
So yeah, just as a time check, it is now currently 1:15 here local in Palau. If we were to leave at this depth, that'd be about a little over 80-minute ascent. So that will basically give us about an hour of bottom time here left before we have to ascend. And so yeah, we'll have to watch that time closely because we'll need to be on deck and secured shortly after 4 o'clock. And then we'll do have a couple plants of sampling on the way up.
Oh, there's a different one. The red one.
Can push in there.
There's another different kind of sea cucumber. There's so many different kinds of sea cucumbers here. So this one is the family Scleractinian. Oh. Oh. This one doesn't want to do crunches like the other one. I really like the little decorations. Party streamers.
Yeah, that's great.
Okay.
And things we have here are scientists ashore. Thanks, Chris Kelly here chiming in. That looks a little bit like a Hansen Otheria. So thank you so much, Chris and all the other scientists ashore. We have a diverse group joining us from Hawaii, Japan, Palau, continental US, Italy, west and east coast of the United States. So thank you so much for joining us at odd hours.
What we got there? Sponge.
This is a Hyalonematidae, a glass sponge. Is this something we'd like to collect, Daniel?
I mean, you tell me, you know this group better. Let me see. Chris is also here. Let's see until it's.
Yeah, well, Chris would be the one who would want it.
He's probably a few seconds behind us, so yep, and he just timed in, so there is a request to collect this.
Okay.
Okay.
And I'm assuming, and I'm going to just confirm this, but I'm assuming we're going to take from the base the entire thing.
Yeah, it'll just sort of come out. We can stick it in the starboard box. Could we also get a zoom on the base? I think there's a little opioid down there too.
Pushing for zoom.
All right, wonderful. So thank you, Chris, confirming that it is like nothing that matches the extensive guide.
Zoom in on the base there.
Yeah, I've never seen one quite like this with the yellow. Was there a tiny cucumber on there? You see the little, little switch?
Opioid.
And there's an amphipod on there too.
That's great.
Okay.
That's like a good view of the spicules. They're like, these are, it's like fiber optic. These long pieces of glass that are twisted together that make up the stem of this sponge.
Come just a little wide. That's good. So, I'm looking to grab it at the base here.
Yeah, she'll just pluck like a flower.
Okay. Snipped it there.
Perfect.
That's definitely floaty.
Yeah.
So in the starboard bio box, we've just got that fish net with no fish.
Fish net with no fish.
In the aft large box, number F, or letter F. And then we have one other sample in one of the smaller bins, so maybe this could go in the forward large bin in the starboard bio box.
Okay. Can hit the sample salvo.
Oh, do you want to secure that thruster?
Yeah. All right, can open the box.
All right, opening. Okay.
Snaggletooth. There we go.
That little piece might get caught, but
It'll be fine.
excellent. Closing.
Yeah, great collection. Thanks, Jake.
Yeah.
That was cool.
Yeah. I was
Okay.
curious how you were going to wrangle that one. That was like a balloon of a sponge.
I
That was
know.
great.
I had no
Sponges
idea.
are always
Just
so
went for it,
It
challenging.
worked out.
Jake's on point this morning.
Probably because I rolled out of bed.
You just roll out of bread and you didn't like put a lot of like too much stress
No,
on
no
it.
coffee. I'm just relaxed.
Yeah.
Megan, can we just get a quick, for awareness, like how far away from waypoint 4 we are, where we
Oh, where we started?
started, just for reference.
Okay. Yeah, you can hit it.
So we started over here.
Want to come up a little bit?
Yeah, sure.
We have gone about over a kilometer. Oh, well, close to 2 kilometers, I'd say.
Excellent. Perfect. Yeah, that's very helpful. Just very also future planning. We're trying to get our pace here right and set up dive plans that are achievable.
Mm-hmm. Yeah, and some terrain is easier to work in than
Yeah.
others.
Yeah, absolutely. And then
It's easy to make tracks here because, you know, no big rocks to worry about.
yeah, and less things to zoom in and yeah.
Oh, it's a purple one.
It's that little yellow guy.
A leaf.
Oh,
Oh, that one's fun.
Yeah, I saw that one on the cinema cam.
The diversity of cucumbers here is quite impressive. So this one looks like it might be in the family Hyalonematidae,
Zoom there.
another type of sea cucumber. It has lots of little pseudopodia as well.
Nice.
We could make a guide to the cucumbers of Palau with all of the ones that we've seen.
It has like spikes, all except for like if you buzz down the middle.
Yep. Just going for a really interesting hairdo, you know?
Just gotta be different.
Mm-hmm.
Weird looking.
Living an alternative lifestyle here in the Palauan Trench
Alright,
areas.
come on. Thanks.
Sometimes I feel like we're playing Pokemon. Gotta catch them all. It's like, oh, I haven't seen that one. Let's add it to my Pokedex. Feel like that's my job.
It's really accurate.
Maybe that's why I loved that game as a kid.
I don't know, the Switch version is pretty good.
Oh, there's a little hermit crab. Right there on the lower right. See it?
Oh yeah, running away.
Run away.
Put it dust it up. Dust it all up. Go for a zoom there. Carrying something.
Oh, it's its shell.
Oh, it's like another little.
Oh, that's its arm.
Oh.
Because they usually have one arm that's larger than the other arm. I think it's got a little large claw.
Huh. Nice.
I wonder if it's a zoanthid on its back or not. It's kind of a weird shape.
All right, ready to move on?
Cool, thank you.
That's great.
Bridge nav. All right, we're ready. 40 meters, 290. Thank you.
Yeah, while we're moving here, I just want to take a moment to acknowledge two key partners in over the years, one and one more recent one. So since 2013, the Museum of Comparative Zoology at Harvard University has been the official repository for biological specimens collected on EV Nautilus expeditions. There's over 4,400 specimens there collected all over the world, and most of them collected because they were thought to be something new to science. And so yeah, it's a tremendous asset and a resource that's available to all our researchers. They can request samples on loan or go visit their archives and study them there. And then this last year we started a new partnership with the Ocean Census project. It's a 10-year project funded by the Nippon Foundation, is trying to accelerate the discovery of new species from past, present, and future explorations of the deep sea. And so we're looking to partner with both of these institutions here in the spring to organize a workshop to basically accelerate discovery from the large number of specimens that have accumulated at the Museum of Comparative Zoology. This will include sequencing of a whole suite of the specimens that have been collected and will continue to be collected, and then also morphological analyses by different taxonomic experts, so stay tuned.
Hey, we have a request for an image of one of these crop circles from Chris Kelly, our scientist ashore.
Okay.
I think I see a good one
Crop
right here.
circles.
I think earlier he was saying that these crop circles could be a result of ecurin being present. Is that's some kind of worm?
Yeah, the echeurin is a type of worm.
Yeah.
They have this really long feeding proboscis that it'll send out, so they'll bury down in the sediment and they'll like send out this proboscis and like drag it back.
Go for a zoom there.
Yeah, this one looks old, like it's been here a while.
Chris Kelly says he has no idea what formed it, but appreciates the zoom.
Okay.
So we're probably all good on that.
What kind of fish was the fish that got suctioned up? Do we know?
From the previous dive?
No, from
There's
the
one
one
in
the one that's in the
suction.
rosette right now.
Yeah,
jar
let me
2
see
or
if
something?
there-
Jar 1 or 2?
I saw it in the
So,
jar. It just
yeah.
kind of was wiggly.
Yeah, well, we have in Slurp 1, we have a sea urchin and a crinoid. And in Slurp 2, we have a shrimp with long tentacles. And some kind of lizard fish,
Oh,
possibly a Bathytyphlops.
ooh, Bathytyphlops. That sounds cool. That's like a blind one.
Yep. That's in Slurp number 3.
I'm happy that the fish collecting is happening. It's really rare that we've been able to do that successfully. And so far we've like done 3 dives and got 2 fish.
Yeah.
Key ingredient to that whole thing is bringing a fish expert on board.
Yeah.
Very
Catch
true.
that fish. Yeah, us coral people are like, ooh, more coral. bridge, Nav.
One of the scientists ashore
Can we move 40 meters 290? Thank you.
is sharing that Martina Gagliotti says that some lizard fishes are known to maybe make these circular shapes,
Oh.
which is kind of an interesting comment. Didn't know that.
I didn't know that either.
I wonder why.
Maybe it's like a way of mating with
Of mating, you
yeah,
said?
yeah. I took this class and they were talking about how some fishes would like draw like little
Is there something in
intricate
the hole?
circles in the sand to attract, and then the female would like lay their eggs in the middle of it,
Oh, wow.
and then the males would go and fertilize the eggs. So that could be like a reason why they do it.
Nice. Good
Push in there.
good comment, yeah.
Yeah.
One of the same scientists
Oh,
ashore.
it's a leaf.
It's a leaf.
There's
It's a
something to the
leaf.
right, though.
Oh, cucumber?
Oh, yeah, it's a cucumber. All right.
This area almost seems like the seagrass of the deep sea, like a seagrass bed of the deep sea, with like all these seagrass and sea cucumbers.
Well, all the seagrass came from shallow water.
Yeah, yeah.
Interesting. Yeah, so continuing the conversation with our scientists ashore, Martina agreed that there is some sort of ritual behind this behavior if it's this kind of lizardfish making these shapes or some kind of fish. And Chris wonders if the troughs are formed by rat-tailed fish with their shovel nose faces designed to dig into the sediment to forage.
Could be.
And we were seeing these dark objects around the crop circle. I think on the last one there was maybe just one that we saw, but they were evenly spaced, and so he thinks it could be a bit strange for debris to distribute like that, so maybe it is involved with some kind of, yeah, some kind of ritual, some kind of mating ritual.
Jake, did you get a sense? I mean, it looks like the current has been coming, or slight current from the south here. Is that right? Just
Yep.
going left to right at screen, yeah.
Yeah, that's right.
Yeah, so we're pointed almost due west right now.
Yeah, this one is a pufferfish. Can't let it. Definitely. Oh. We might have a request for a zoom on this crop circle.
Yep. Push in there. That is weird.
Interesting.
This one looks like an old one. It's
Yeah.
been here a while. And it'll collect some debris because it's a little bit lower than everything else.
Can we zoom any more? Yeah. So Chris is convinced that's the Ikurin. Oh no, he says not.
I think it's made by something that's living in the sediment. But that's my thought. I think that should be good.
Okay. Moving on.
Just as a time check, about half an hour of bottom time left.
Bridge nav. Can we move 40 meters to 90? Thank you. Jake, we kind of want to look at this little thing that we just passed.
You want to look at it?
Yeah.
Yeah, right there, bottom of the screen.
There's a little something something at the tip of it.
All right.
Yeah,
Push in.
Connie spotted that nicely.
It's a worm tube.
Yep.
Oh, and I think the worm was out, but then we scared it, so it went back in. Okay, thanks.
So the tube is like a part of its morphology?
So the worms will make these tubes, and they live in them.
Oh. Cool, cool.
Yeah, just as a look ahead here for folks that are joining us, so we are going to be leaving the seafloor in about 30 minutes from now and making it's going to be about an hour, 40 minutes of ascent to the surface, securing the ROVs on deck at 4 o'clock local time. And then we'll have about 24 hours of no diving. We'll have a personal transfer to complete and then a long transit out to the easternmost extent to the Palau National Marine Sanctuary. There's a seamount on the very far eastern corner of the Palau Exclusive Economic Zone, and that seamount has not yet been mapped. And so we'll be mapping that overnight and then picking a ROV dive track from that mapping data. And so yeah, we'll expect to dive there about 24 hours from, well actually 26 hours from right now. There'll be another 12 to 14 hour dive, and we'll continue this sort of cadence. So we've been picking of diving 12 to 14 hours and then 6 to 8 hours of mapping and transit, and we'll just rinse and repeat here through the end of our first leg of the expedition that will end on November 8th. Then we'll have 2 days in port, and then heading back on November 11th, and that will be an 11-day leg. So a lot more exploring to go. And also for our scientists ashore that have been following along with the dive plans and situation reports, all the dates and time that we share, and those are all in local time, so in Palau time, so we are across the date line, and so you should see dates that are 1 day later than your equivalent date, and that's a little bit of a difficult mental exercise. Most brains are not wired for that, including mine, to do that on the spot, do those translations.
So a question for the science team. A viewer wants to know, how many samples have we collected so far, and how do you get them safely from the ROV to the ship?
So far on this cruise, we have collected 43 samples, but for each one of those samples, we have additional subsamples or associate organisms. So we may intend to collect one thing, but there might be something else that is living on it or with it, or living on a rock, and then we have both a biological specimen and a rock. So I'd have to double check on how many. I don't have that number handy right now, but I believe at least double that in total number of samples plus subsamples. And then we would also have an additional number of surface eDNA samples that aren't even included in that. So we have quite a lot of samples so far, and we're just a few days in. The end of a cruise, we can end up with hundreds of samples, including subsamples. And the other question?
Oh, how do you get them safely from the ROV to the ship?
Well, the ROVs are actually attached to the ship, so so there's a process of recovering. Yeah, there's a process of recovering the vehicles onto the ship, and once they are on the ship and safely secured, then the science team moves the samples from the vehicles on deck into our wet lab.
Go for a zoom there.
Spiky hairdo. Reminds me of those water toys.
Yeah.
That's great.
All right.
So, so another question. A viewer wants to know how far to the rocky slope.
Oh, well,
Yeah,
I mean, probably pretty far.
yeah. So we are about 3 nautical miles now from the edge of the reef, and I actually don't know if it, I mean, the slope is kind of, this is the same kind of slope throughout. If you look at the height back here, maybe it gets a little bit steeper further up. But it could well be this, yeah, it could well be this similar terrain, just getting shallower and shallower. And it's been pretty consistent in terms of this type of substrate.
That's a fun looking sea cucumber.
Go for a zoom.
Oh, it's just a short.
It is really,
I
it's
know.
really cute morphology.
It looks like a little rabbit.
All right. Continue on.
It's just so round.
Another Nematiad there, just passing on screen. Oh, can we take a quick zoom here, bottom of screen?
It's like the one we picked up before. Yeah, the pink synallactid. We got one of those in the box.
Perfect.
That was the one that tried to escape.
Perfect. All right. Excellent. Thanks for confirming, Megan.
Now it's gonna take off, maybe.
Do it.
Oh,
No.
Oh, it's a different kind of show.
Plane dead.
Yo, it's fine.
Is that some detritus there? Yeah.
Yeah, it's like more plant matter. Bridge nav. And we move 40 meters to 90. Thank you.
Maybe a fresh one. The mound. This is like what we've been seeing, but
No,
fresher.
the one that they were looking at was like had the holes all around the outside.
Okay.
Yeah, that's great.
Yeah, but there are definitely some animals that make mounds.
Do do sea cucumbers make mounds?
No, no, they just meander across the seafloor, and they'll leave these little castings along. Sometimes they're like little squiggles or long tubes, or, you know, they're very distinctive. With how many sea cucumbers we've been seeing, I have been seeing a lot of that, but most of the ones that we have been seeing do like to swim. So a lot of times they'll drop a payload before they take off to go to a new area. Bridge nav. Can we move 40 meters to 90? Little hermit crab. Yeah. Yeah, they're quick. Oh, that one's fun. It's like a party.
Over zoom there.
You
You can see the little trail it's left.
Yep.
It's so pretty. It's a good color.
Just tumbling. Is that related to the sample we got?
Yeah, we in that same family we had several Nematides. Yeah, it looks like just about 10 minutes of bottom time left. I have an 82-minute ascent from this depth.
Oh, there's another one of those little short ones. I love their vellum, that like little sail thing it's got going on.
Push in there.
Don't you want to just like squish it a little bit? I feel like it'd make a really good plush. Maybe I'll crochet a bunch of sea cucumbers.
That would be amazing.
We have a lot of options.
Mm-hmm. Oh, I know. I lost track of how many different kinds we've seen on this watch.
So we have a couple of notes from Chris Kelly, our scientist ashore. One was telling Megan not to play with the sea pigs, no poking. And the other one, I'm not sure if we have time or what you think about this, Daniel and Jake, but he's wondering about digging maybe into one of these crop circles to see if we can see the organism.
Okay.
What do you think about that?
Yeah, I can. I mean, it is
I think
a very
it's going to be
abundant.
real dusty.
Yeah, I think we'll lose sight.
Yeah, it'll definitely be dusty. But
I'm not sure if we will really find anything. It might just
Probably
burrow.
not. Yeah, the thing
It'll
will probably just burrow
away.
down.
Yeah. I think we just passed a crinoid, right? Or what was that?
Yeah, there is a crinoid and Cerianthid. I'm glad we saw that sponge. That was really cool. I thought we'd see more of them.
yeah, one lonesome sponge.
I bet there's more. We just chose the wrong path to see them.
I think it's really neat when we recover and to watch you guys remove them and actually see like how large
Oh yeah.
some of these things are.
Or how small they actually
Yeah.
are. Like on the screen it's like, oh, it's huge, and then when you get it in the lab it's like, oh, it's like 2 centimeters.
Yeah.
Oh. Is that a little radial kind of crop
Yeah,
circle?
that's what the echurans do.
Yeah,
they
exactly.
make those like spoke trails.
Yeah.
Oh, what is this guy up ahead?
Oh.
Don't tell me it's a big leaf. It's not going to be a big leaf.
No, that's definitely given cucumber.
A healthy cucumber.
Oh yeah, that's a really nice healthy one. Like a benthidites.
It's like the
This is a new one. We haven't seen this one yet.
Wow.
It's like the sweet potato of cucumber.
Kinda. It's like a little bit lumpy, but in the best possible way. Some Ride talks about them in terms of mouthfeel.
Push in.
We should compliment people like we compliment sea cucumbers.
Yeah.
Yeah.
It's like, you're just the best color, and you have such great texture.
I like that specifically. You're a little bit lumpy, but the best
I
kind of lumpy.
I feel like
the best
this
kind of lumpy.
sea cucumber has like an old soul.
Yeah.
Well,
Seen
looks
some
like
stuff.
a brain almost, like the texture.
Mm-hmm.
This is a big sea cucumber.
Yeah, it is.
I want to get the lasers on it.
Well, it's turning for its glamour shot. Are you gonna do crunches for us?
Yeah, I'm just looking at it compared to like the porch.
Mm-hmm. No.
Oh
the
wow,
front
that's
of the
a huge
basket.
one, isn't it?
They get
Yeah.
really big.
It's like 10, 20, 30, 30 centimeters.
Oh,
Oh,
Oh, he's mad. Get those lasers off.
well, he's ready to do
Oh,
some crunches.
He is going to the restroom. That's what's.
You gotta lose some ballast if you're gonna
Yeah.
swim.
Oh,
Mm-hmm.
yep.
We're going to get taking flight.
Oh, what's happening?
just cleaning the sediment.
So
It's
graceful. I love
Very
it.
graceful.
So neat to see, though.
The cucumber probably feels so violated.